stroke, should predict the prognosis, facilitate therapeutic stratification, therapeutic monitoring. It should indicate whether the patient is going to have hemorrhagic transformation or not. It should have like a stroke clock, like we were discussing, that it could be up to 1 week also. So you have a clock where you can increase the number of patients and receive them. It should facilitate directing the patient to a hospital where anything can be done. It should be able to subtype diagnosis, and should it be a target for treatment also. If you are asking for a dream biomarker, we don't have, that's why we don't have biomarker in clinical practice. So it is as simple as this. Well, you all know this pathophysiological cascade of stroke, the inflammation, and probably the source, the thing which Robert was telling, DNA. I picked up from there, self-plasma DNA. You have specific biomarker either coming from neurons, from oligodendrocytes, from Gallagher cells, endothelium, or to hematopoietic blood cells. But something which is common to all of them will have necrosis or apoptosis, and they will release self-plasma DNA. So, self-plasma DNA, it can have early rise even before neuronal necrosis or apoptosis, because it is a very short half-life. Self-plasma DNA, we all have DNA inside our cells. Once it is released from nucleus outside the cytoplasm, so these are fragments which are roughly 160 base pair size. These are short fragment of double-stranded DNA which are into circulation, and they have a half-life of 5 minutes to 150 minutes. So self-DNA, practically it's a snapshot of dying cells. Cells are dying, there is stagnation, so it will give you an idea what is actually happening inside the brain. So this is how it is. So as I said, start rising even before, when there is ischemia, you have a heart carotid plaque or something in your heart, there will be neutrophils around there, and this is a major source of, these hematopoietic cells are major source of DNA in normal human blood also. But when there is inflammation, it will start rising. So, and there is something called necrosis. You have these neutrophil extracellular traps where you have self-plasma DNA. So these are the self-plasma DNA fragments which are, as I said, small, 160 base pair size. Why this is unique? How we can measure it? The size is such because of its nucleosomal packaging. So it is to be same, whether it comes from neutrophil or macrophage or from neuron or from endothelial cell, wherever it comes from, whatever the source is, the size would be same, so that you can detect it in blood. Well, as I was telling, a wonder biomolecule, self-plasma DNA as possibly what happens, this is a kind of family of DAMs, damage-associated molecular patterns. So you have pathogens, bacteria like what we call PEMs. So many ATP, protein, cell-free DNA, so this acts like a DAM family of protein. Because DNA is bound to protein, that's histone. So what happens here, the self-plasma DNA, this is recognized by pattern recognition receptors on the cell surface. Primarily, this DNA sensing is done by 3 receptors, tolerant receptor in the cells, then you have AIM2 and STING pathways. So basically, this itself activates these receptors and transcribes many pro-inflammatory cytokines, interleukins, etc. So what happens? It drives inflammation. Is a biomarker, it is driving inflammation also. Once there is inflammation, there will be more cells will be recruited there. Again it is driving, so it's a kind of amplification of the cascade. So this is how cell-free DNA will rise. You can target this in the very beginning, maybe like we use TPA, you can give DNA which is commercially available. So these DNAs can be given so as to blockade this cascade at many steps. So there are many checks here. You can right away straight the entry, because DNA is present in blood circulation which removes self-plasma DNA, and that is why half-life is as short as 5 minutes to 150 minutes. And in lysosomes you have DNAs too. So you can use DNA as a therapeutic agent also. And there are various other therapeutic agents which I am not going to detail of those. But since I found that inflammation is a major key factor for neuronal damage, so this is, since we cannot have drug trial, anti-inflammatory very easily available in our country. So we started using curcumin in some subset of patients, and this is as an anti-inflammatory. So this is already, these are published data. So it does improve outcome primarily because of strong anti-inflammatory action. There are various types of stroke biomarkers, just to recapitulate. Biomarkers of brain injury, S100B, MLP9, neuron-specific enolase, glycoprotein acidic protein. Then you have biomarker of inflammation, like interleukin-6, TNF-alpha. You have biomarker of oxidative damage. You have biomarker of lipid-related biomarkers. You have biomarker of thrombus formation, like D-dimer. You have cardiac biomarker also, like BNP. But you have different biomarkers. Cell-free DNA is the only thing which is common to all of this. So it falls in all 6 categories. Well, so this is what it is, how we started doing that. This, once you centrifuge this blood sample, there is something called plasma, which is here as a supernatant. Then you have a buffy WBC coat and there are RBCs on the bottom. So, cell-free DNA, if you can detect it, you can probably do early diagnosis. Depending upon the higher level, you can prognosticate whether the levels are rising or falling. You can predict disease progression. You can optimize your line of treatment, MT or TPA. Response to treatment, whether it is falling in response to your treatment or not, or you can target it as a treatment also. It will reduce post-stroke opportunistic infections. So how does it happen? Because there is a massive release and there is a massive inflammation in the body, you are in a transient state of immunosuppression. So that's again one point which I need to highlight. The work on cell-free DNA started right back in 2003 when Rayner published that it reduces 6-month mortality, higher cell-free DNA, higher 6-month mortality. Then outcome study, another study which established that you have poorer outcome if you have higher cell-free DNA level. And then on DI-based study also, this was our own study which we published in 2018. So the whole idea was here, when I was doing mechanical thrombectomies, I wanted a marker which can actually, give me an idea whether my patient is going to be benefited or he is going to have hemorrhagic infarct. So we did his first study and published in 26 patients with proximal MC occlusion. We took 5 ml of blood sample within 6 hours. They underwent standard laboratory imaging workup. We collected this 5 ml sample and this was centrifuged and 1 ml of plasma was extracted. So basically the cell-free DNA takes like one step is, first of all you centrifuge that, extract nucleic acid or DNA with a kit which are available. It takes around 30 to 45 minutes to extract that, and then you measure it on nanodrop, or I'll tell you the material also later on, and then you do RT-PCR. So this process itself takes 5 to 6 hours, which is actually not possible in clinical practice. Having result in 6 hours, then it's probably a futile thing to do. Well, since this was part of research study protocol, so we found that it is significantly related with your NIH stroke scale recognition and 3-month outcome by MRS. Patients, those who had less than 10,000 kilojoules per liter, had better outcome whether they were treated by mechanical thrombectomy or TPA. So this was initial study published. As I was saying, the steps in extraction, one is extraction. So you have commercial these nucleic acid extraction kits available in lab, which takes around 45 minutes to extract it. Then you measure it with an instrument called nanodrop or a qubit or a tap station. So you assess purify and then you subject them to RT-PCR and come what amount is there, you know that. We expanded study from here on. Then we went up to 54 hours, results were same. And this was last year publication, where we did it almost in 188 patients. Here we extended window period also. We went up to 24 hours because at that time Dawn trial was already published. So we also extended window period up to 24 hours. The problem with cell-free DNA is it takes 5 to 6 hours to measure it. So we were looking for a solution that can we reduce this time, because it appears to be a novel biomarker, but can we reduce this time? So solution was to develop a radiogenomic model that can integrate cell-free DNA along with imaging variables and other scales. And the second thing, to develop a POC, point of care type testing. So this was the whole idea. So when we were doing it over the years, we realized, see, I told you there are 3 steps in the beginning. Extraction, first thing is, this is the extraction kit. This is a nanodrop which measures cell-free DNA, it assesses its purity. This is a nanodrop. From here to here, it takes only 45 minutes. Then with RT-PCR, you actually amplify and quantify it more precisely. So suppose you omit this step. Here, from here to here, you can just measure it on a nanodrop. You just measure it on a nanodrop. Only thing is calibration and standardization of the nanodrop should be good. Nanodrop is a very simple device present in most of the lab. It gives you label of cell-free DNA within seconds. Only thing you need to have purification. So what you do is here, because cell-free DNA gets absorbed, it's a spectrophotometry. It gets absorption at 260. You have protein also in your blood, in your plasma, fibrinogen and other plasma protein, which are absorbance around 280. So if your ratio of 260 to 280 is 1.8, you are measuring probably pure cell-free DNA. So this was the thing, when I look back, we went back to our data, we found 50 patients where we had, out of these 188 patients, the calibration was perfect and we measured cell-free DNA on a nanodrop, which is again nanodrop will give you result within half hour or 45 minutes. So this was interesting, and based on that we did healthy individuals like 1 to 10 nanogram per ml, minor stroke based on NI stroke scale 10 to 20, 20 to 40, and if you have severe stroke, these are more than 100. When your NI stroke scale is 21 to 40. So you can actually classify severity of stroke based on cell-free plasma DNA level, which is, just to repeat, it's a snapshot of dying neurons in your brain. Well, next is developing a point of care type of testing for this. So that will give you quicker results, like a pregnancy kit, like a urine albumin analysis strip. So the problem here in assessment of cell-free DNA is you have to centrifuge it many times. When you are doing either nucleic acid extraction or when you are doing RT-PCR, you have to repeatedly, every centrifugation would take 10 minutes or 15 minutes, and you actually consume a lot of time. So something that so if you bypass this step of centrifugation, so there are like newer things, microfluidics, which this is a microfluidic system. So you actually skip this centrifugation part. This works on capillary action only, and there are various things like sample, washing buffer, elution buffer. When you just add to this chamber, it goes to silica membrane, and it gives you quicker results. So this is one thing which I just read this in literature. We don't have that. Then the future belongs to probably DNA chips. Nowadays you have DNA chips, DNA sensors. This could be a point of care testing kind of system. So it could have like this platform, DNA sensing platform could be either a microfluidic device, it could be a lateral flow assay device, it could be sometimes paper-based analytic devices, like an organic paper we use, for making craft and those things. So this is the kind of equipment a DNA sensor would be. You have capture probe inside this. So you have, this is the platform which will sense DNA, you have capture probes, and you have various detection techniques. You can do it by color change, colorimetry, electrochemistry, electrophoresis, or by fluorescence measurements. Well, this is something which is available, but not for cell-free DNA. So here what you do is lateral flow assay, it works on capillary action. You load the sample here on a conjugate pad, and through capillary action it moves over a nitrocellular membrane. Like in pregnancy kit, you see this is a strip configuration. So this is a strip configuration. You have a control line. If you have a test line positive, target sequence is there, the capture probe is there. You see two lines, it is positive result, and further you can quantify this also as well. Then comes this paper analytic devices. So these are the paper, simple, simple organic paper devices. You have fabricated this paper by wax printing, wax tipping, many other methods. You provide some hydrophobic wax barrier, hydrophilic zones. So basically what happens, once you load the sample in the pad, liquid moves through paper using capillary action. So there is no need for centrifugation, no need to pump or any other extra equipment. You just have a paper device in your hand. There are various microchannels inside this paper, and this works on capillary suction. And you have short DNA probes at one place in these devices, which are short DNA strands that will bind to your targeted analyte, that's cell-free plasma DNA. Various nanoparticles in picture. So this is the advantage of using this DNA. This probably could transform healthcare in times to come. The key advantage of POCD devices for cell-free plasma DNA are speed. You can result within minutes rather than hours. Accessibility. It is something which can be used in remote, resource limited areas. So the paper device can be used in any village or any remote area in the country. Obviously, there are various limitations and challenges to this, like specificity. Of course, we are not talking something specific here. We are talking something very sensitive here. So specificity per se is not a problem. You have already assessed your patient well. Clinical history, imaging, and everything is already in your hands. Obviously, it needs to be standardized. And in future, you can use multi-omic integration, combining cell-free DNA with protein or microRNAs or metabolites, other protein metabolites or compounds. Yes, sir. This is by last time. AI-driven analysis, machine learning can give patterns, and based on unique methylation pattern, you can even diagnose that whether it's a cardiovascular stroke or a carotid plaque, origin homolog, or somewhere else, because it can actually pick up the cell of origin also by unique methylation pattern of cell-free DNA. That can be done. I think that's all I had to share today. Thank you very much.
Thank you, Atulabh. Cell-free DNA from the plasma. I didn't get at what time you collected the blood sample from the patient. Was that before thrombolysis started or after thrombolysis? What was the time frame that you got the blood? And also, if you collected the blood at later time points, would the cell-free DNA levels drop or were they still elevated?
Yes, that's very relevant question, sir. We took blood sample right at the admission. Yes, at the admission. In the emergency itself, we have collected this 5 ml of blood.
So before license?
Before anything. Before the patient should get on CD or well. So part of that's a standard, we took it in the emergency suite itself. Because you are very right, because what time you collect cell-free plasma DNA will give you different value. Okay, so 24 hours, it depends if you have a small or neural injury. It will, as I said, it is a snapshot of dying cells. So if you, depending upon your neuronal blood front, which is massive or progressive, it will behave. So that's how, and obviously yes, you're right, it peaks within 24 to 40 hours, unless until you have ongoing ischemia and neuronal necrosis. Usually, after 48 hours, it will start dropping. And by the end of 7th day, you will have negligible levels strength.
Thank you very much. Thank you, Atulabh. Thank you, Robert.
Thank you so much. And if you look at the time, it's exactly 6:30 and we will...
Actually, fantastic work, I must say. What I wanted to ask, can you distinguish hemorrhage from ischemic stroke to this?
Sir, actually, that's something, it's, I mean, in my study, it was not there. It could not differentiate between ischemic and hemorrhagic stroke to be very clear, because, it was in literature also, I tried matrix metal of MMP9. So that is something which is described in literature, which distinguishes between ischemic and hemorrhagic stroke. But in my study, I did these S100B and other things also, but I could not find any, label which can cut off between hemorrhage and ischemic stroke.
There was a recent study of gliofibrillary protein, which can distinguish between, and also its point of care, you can distinguish between hemorrhage and infarction. And that is very useful for out of hospital thrombolysis. You can distinguish without doing a CT scan. So this cannot. or you haven't done the study?
No, sir, I have not looked from because I think stroke primary imaging would not be replaced. So anyway, patient has to undergo a CT scan. So I did not actually try to look from that point of view because every remote area nowadays in our country also would have a plain CT at least.
Last comment from Dr. Robert.
Thank you for that.